Hello, in this video we will discuss about the DNA replication with detail. So the DNA replication is done in a three-step: initiation, elongation and termination. Initiation step is basically we will discuss first is the initiation protein involving during initiation. So the first is the ORC is the origin replication complex which that is bound with the ORI site. ORI site in the DNA where the replication bubble will be occur and the replication will begin. So there are many origin site which that is binding the ORC. On the other hand, the CDC6 is the cell division cycle 6, basically is the helicase loading for using the helical structure DNA unwinding. So the CDT1 is basically is the CD10 dependent transcript 1 which that is used for the helicase adding and basically the interact with MCM ring. So the MCM ring is basically the 2 to 7 monomeric protein. This monomeric protein is basically mini chromosomal maintenance with the helicase. And CDK and DDK is basically used for the kinase protein. This is and cyclin dependent kinase, BBF4 dependent kinase and initiation protein regulation. This is used for the initiation protein regulation. And on the other hand, the CDC45 is basically cell division cycle 45 and interact with MCM7 and that is the basically polymerase alpha. On the other hand, it is also polymerase alpha. So also is the interact with the MCM7 2 to 7. So the genes protein is also which that is the co-activator of initiation and this is the MCM assembly. So the next is the PPA which that is used for the replication protein A. It's used for the prevents re-annealing of DNA strands as SSPS. So basically is the SSPS like a single stranded binding protein is used for it. On the other hand, the RFC is the replication factor C is the DNA clamp load and load pCNA. This pCNA is used for the basically we will discuss, and the polymerase alpha is another is basically is the initiate DNA synthesis on primer and DNA polymerase delta is basically leading and lagging strand synthesis. And the primase is the binds primer attached. So the primase is another protein which that is binds primer and modified the primer for the specific site. So let's begin to understand with the mechanism with the attachment of this protein and initiation factor to initiate the DNA replication. So this is the DNA replication factor for the initiation of the DNA replication. So here is the ORI site which that is the DNA replication bubble will be occur. So the ORC will be bound with the ORI site and after the binding it will be MCM ring will be bound with during the G1 phase of the cell cycle. So that CDC6 and CDT1 will be bound by the recruitment of the ORC. So the ORC will recruit the CDC6 and CDT1. After that binding with the and also bind with the MCM ring. This MCM ring contain a 7 monomeric protein. So on the other hand, after this basically after the MCM ring binding basically is the ORC will be bind with the protein was CDC6 and CDT1 which that is also recruit the helicase by the binding of the MCM and CDC6 phosphorylase to excrete or release from the ORC site and ORC will be remain. But here is the CDC4 4 to 5 will be recruited by the phosphorylation of the MCM. So the SLD3 to 7 complex will be formed and genes will be recruited by the MCM by the phosphorylation and the CMG assembly formation. This CMG is the basically CDC4 to 45 and M mean the MCM2 to 7 and genes is the G is the genes protein. This is the complex formation for the replication during the initiation. So the ORC is the remain and CMG assembly complex formation. Basically the CDC 45 and MCM as well as genes complex formation. In this CMG, basically the CDC 45 and MCM and genes formation, which that is the complex is used for the initiation, and that is also recruitment of RPA protein and polymerase alpha and primase. RPA basically is the RPA that was RPA, which that is replication protein A, which that is prevent the re-annealing of DNA strands as a single-stranded binding protein. So here I am drawing the during the replication. Basically the replication bubble will be formed. So I am drawing the side of the where the replication will be occur. So here is I am drawing the structure of the DNA during the replication. So here is the structure of the replication where the replication fork will be occur due to the initiation factors. So the DNA replication bubble will create the two replication fork. So I am drawing the single replicating fork to understand. So here is the helicase enzyme which that is used for the double helical structure to unwind the separation of the two strands. And here is the topoisomerase enzyme which that is lead to topology of the DNA and remain the shape of the DNA will maintain. So here is the basically the single stranded binding protein will be attached, and here is the replicating fork will be occur, and this here is basically the single stranded binding protein will be occur, which that is maintain the structure of the single strand of DNA. So here is another is basically the primase. This primase is synthesis of RNA primers and short RNA molecules that act as template for synthesis initiation. So during the two strand will be occur, leading strand and lagging strand. Leading always the DNA replication begins always 5' to 3'. So that is the problem. Basically the one strand is 5' to 3' direction, while on the other hand the antiparallel strand is 3' to 5' direction, which that is antiparallel to each other. So DNA's double helical structure is antiparallel. So here is the leading strand and lagging strand. Leading strand is simply replicate and their primer bind after the primase enzyme and primer binding to begin the polymerase chain reaction, which that is a polymerase DNA polymerase delta will bind to initiation and elongation process. So this is the elongation. While here is the lagging strand, which that is problem. Basically the lagging strand is always 5' to 3' direction, but here is the near to the replicating fork will be bind the primer and begin the polymerization, and DNA polymerase 1 will be alpha will be bind and to annealing process, and the primer will remain here, and there is a different segment will be bind and the DNA polymerase 3 enzyme, which that is polymerized. And here is the basically the segment will be left behind. That segment is known as the Okazaki fragments. And there is the many gaps of the 200 to 1000 base pair, basically gap of the nucleotide will left behind, which that is again transcribed or annealed by the DNA ligase. not transcribe but read. And DNA ligase enzyme bind, and where the site of the DNA ligase will be bind and addition of the nucleotide. On the other hand, the primer will be released. After the releasing of the primer, Okazaki fragment will be occur, and that is the DNA ligase enzyme used to ligation of the strand, and complete the strand will be occur rather than the segment will be found. So that was the basically overview of the DNA replication, which that is the replication fork will be occur, and that is the lagging strand problem, which that is the 5 prime to 3 prime direction, but the 5 prime to 3 prime direction is basically is occur when the 3 prime to 5 prime direction is not possible. So here you can see that this primer will be bind, and here is you can see that a leading strand is directly and continuous, but the lagging strand is discontinuous. So in this problem, the discontinuous manner of the lagging strand will be read by the Okazaki fragment will be formed, and this Okazaki fragment will be DNA polymerase enzyme will be cover this Okazaki fragment, and DNA ligase enzyme is here will be add. This DNA ligase enzyme will be covered this segment which that is empty segment and not read still. So here is the leading strand is directly by the primer binding, and polymerase delta will be reading and complete the formation. While on the other hand, important thing is that I will discuss about the near to the replicating fork is here. Basically here is the replicating fork, that is a many problem, which that is the telomerase enzyme will be solved. We will discuss in later in this video. So first of all, I want to explain the DNA replication begins always 5 prime to 3 prime direction with the detail. So here is the nitrogen base and deoxyribose sugar and phosphate group. This is the nucleotide, and this nucleotide is contain carbon number 3 containing a hydroxyl group, and this hydroxyl group is known as the carbon number 3 is known as the 3 prime site. So the hydroxyl group, a 3 prime carbon containing hydroxyl group will be bind with the phosphate group in the next nucleotide. So it means the 5 prime direction to 3 prime direction DNA will be replicate. So this replication will begin to parental DNA is the 5 prime to 3 prime direction, but the 3 prime to 5 prime is the reading replication. So here is this manner you can see that the 3 prime to 5 prime 3 prime to 5 prime direction, but their reading is the basically is the 5 prime to 3 prime, basically is the backward attachment. So here is you can see that is the 5 prime to 3 prime direction and no problem in the leading strand, but the lagging strand can be problem. Why? Because the lagging strand is attached with a different primer to process by the 5 prime to 3 prime direction, because the parental DNA parental RN strand is a 5 prime to 3 prime direction, so it is very difficult to anneal and difficult to annealing, and the primer will be bound with the 5 prime to 3 prime direction with a segment wise. So this segment will clear by the DNA polymerase enzyme, and this polymerase enzyme and the Okazaki fragments will be occur, and this Okazaki fragments is due to the releasing of the primer. So after the releasing of the primer, the Okazaki fragment will be covered by the ligase enzyme. So this ligation will be occur. But important thing is that near the replicating fork, that is the problem, which that is the hydroxide group will be bind and bind and bind again and again, but the still last point basically should be a 3 prime. So that is very problem is that the exonuclease first of all will release the primer. after the releasing due to the exonuclease, so that is the exonucleation will be, we can say that. After the exonucleation, we can say that the DNA ligase joins and DNA fragments together. After the together, the DNA fragments basically is the leading strand is done, but the ligase enzyme will be cover the Okazaki fragments, and that is known as the ligation. Basically, is the lagging strand, which that is a ligase enzyme used to ligation and cover the fragment. That is the fragment is known as the Okazaki fragments will be covered by it. So I hope you make sense about that. But the replication end process is very it's complicated, and the replication end problem is occur, which that problem we will discuss and we will solve by the telomerase enzyme. So the telomerase enzyme is the basically replication and problem solving process, which that is the telomerase activity we will discuss about. So the telomerase is the enzyme which that is adjusts the telomere of the DNA of the chromatin, which that is the chromosome is contained. A telomere is the head of the chromosome. So here is the RNA primer will be released after the replication, and the RNA primer will be when released. So the GTT, GGGTTGG is the sequence. This sequence is basically the telomeric sequence. This telomeric sequence when the release of the RNA primer, so the parent DNA one strand is here, and here is the daughter strand will be occur. So here is the RNA template are present in the telomerase enzyme. So the telomerase is made up of the ribonucleic acid. So the basically the ribonucleic acid containing basically here is the template, RNA template, which that is used for the specific site where the binding is reliable. For example, the CAA ACC, which that is bind with a specific site, so it can be a specific. So this enzyme will be enhance their structure, and here is the bind with the two nucleotides pair. So the pairing of the two nucleotides, the synthesis of parent DNA elongation. So the DNA parental DNA will move to left side. So the left side movement of the left side. After the movement of the left side, basically the primer formation will be occur. This the primer formation is due to the primase, and this is the provided by the telomerase enzyme. This telomerase enzyme is cut the telomere. So the basically is the here is you can see that is the primer will be bind, and that extension will be occur. So this extension is against the GTT GG. So this is in the form of primer. So the telomerase enzyme is provide the primer in the telomeric part. So the telomeric DNA part, which that is provide the primer. After the providing of the primer, basically the GTT, GGGTT, GGGT, GTT, GGTTC basically is the reading, and the against this the antiparallel strand will be occur. Anyhow, here is the primer will be bind. After the primer, polymerase 1 enzyme will be polymerize this and nucleotide bind. But important thing is that this primer should be released due to that this is the primer, and this primer is the RNA primer, and it's basically is contain basically also uracil, and that will be released. After the releasing of this RNA primer, releasing of the RNA primer by the nuclease enzyme, so the our hang flap will be occur. This our hang flap will be cut by the restriction endonuclease enzyme. So this cutting is the basically is the cutting of the enzyme is the telomerase enzyme. This telomerase basically is the telomerase enzyme, which that is cut the telomere part, which that is left behind is the here is the GTTG, which that is activity like a restriction endonuclease enzyme. So basically is the releasing of the segment. After the releasing, every division of the cell will be cut this segment GTTGGC. So this cutting mechanism is very important because it is prevention of the prevention of the extra DNA, which that is overhang flap. So this basically is the overhanging will be released, and here is the each division will decrease the telomere. So this is the telomeric segment will be cut in each division. So that means telomerase enzyme is very important to extra telomere will be cutting. But that was is the extra, but it is extraordinary. So remember about that. It will be increase the aging. Why? Because the shortening of the DNA is decrease the gene activation is also. So here is the termination process will be occur. We will discuss in the last step. So that is here is the lagging strand, and here is the leading strand, and the center is the termination point. But here is the replication is occur, and here is the leading strand. Here is the lagging strand. So here is the MCM 2 to 7 is the ring-like protein will be bind to side, and during the termination point, here is the termination point, and that is the basically the leading strand and lagging strand, which that is here is the SCFE3, which that is the ubiquitin ligase complex. This complex is bind, and the CSF SKP1 CDS3 and Culin and F box is the complex formation, which that is basically you can see that here is the SCFE3, which that is a ubiquitin ligase complex will be formed, which that is bind where the site of MCM and MCM2 proteins 2 to 7 monomer, which that is bind, and is basically is the SCF is the another protein which that contain SKD1 and CDCS3 and Culin 1 and F box basically is the binding site. So here is the leading strand and lagging strand will be occur. So that is the basically the MCM7 is ubiquitinated, and after this the polyubiquitinated with K48 protein. So here is the ubiquitinated is looking like this, and this the P97 remodeler protein which that is add, and P97 and VCD is segregated enzyme, which that is segregation. After the segregation causes the disassembly of active helicase, and this causes the disassembly of active helicase enzyme, and that helicase enzyme will be removed. After the removing of the helicase enzyme, the DNA will be DNA will be wind from the unwinded. So the CMG is here, which that is complex is the disassembled. Basically the CMG is the complex which that contains C is the CDC45, and M is the MCM2 to 7, and the G is the genes protein which that is used for the initiation. So after the initiation, elongation will be the termination process will be terminate this complex and disassembled. So this disassembly of the CDC45 and MCM2 to 7 and genes protein which that is disassembled, and the DNA replication will be clear, and the cell will be divided, and after the division of the cell basically is the cell division will be occur. So that was the DNA replication video. I hope you make sense about that. Basically is the G1 phase and S phase and the G2 phase, which that is the cell cycle. All things is the occur in the DNA replication. So thanks for watching. Please make sure to subscribe, like and share this video, and give the feedback about this video, and I will make another video about the replication. So thanks.